Hello everyone, I am going to explain you the DNA replication in this video. We will study about how the replication taking place in DNA to make its copy. Central dogma you have already studied in my previous class, that how the DNA makes autocatalytic function and synthesize another DNA for the cell division or formation of new cell, or it undergoes transcription for protein synthesis. So that is heterocatalytic. So heterocatalytic in next class we will study. Here we will study about autocatalytic function of DNA, that DNA replicates to form another DNA copy. So here try to understand the machinery required. DNA dependent DNA polymerase catalyze the reaction, polymerization of this replication. Secondly, this polymerization of denucleotides is very efficient process catalyzed by DNA dependent DNA polymerase. You know, enzyme plays very important role. So DNA dependent DNA polymerase, never forget, catalyzes the reaction, and polymerization of the nucleotides is very fast. When we come to the study, it is revealed that in Escherichia coli, having the 4.6 into 10 to the power 6 base pairs, adenine, thymine, cytosine, guanine base pair, adenine, thymine 1 base pair, cytosine, guanine 2 base pair, like this. So many base pairs within 38 minutes. So when per base pair it is calculated, we come to this data that 2000 base pair, adenine, thymine, cytosine, guanine, like this per second. Imagine, per second, 1, 2, 1 second has passed. So in 1, 2, 2000 adenine, thymine, cytosine, guanine, adenine, thymine, cytosine, guanine. Such a fast process has completed in this replication fork. I am going to explain you later on. It's a very expensive process. As we start here from the efficiency, the fast mechanism of DNA replication requires also a fast or very high amount of energy, as well as it is a semi-conservative nature. So half of DNA comes from the parental, but half newly from the nutrition from the cell system it requires. So here nucleoside, deoxyribonucleosides, triphosphate play very important role. Yet you have studied that ATP is the source of energy. No, not only the ATP is the source of energy. It's other three brothers. Thymine triphosphate, ATP, adenosine triphosphate. Here it is thymidine triphosphate, cytidine triphosphate, and guanosine triphosphate. So these all four serve dual function. One, it provides the raw material as a sugar phosphate base. It's a nucleoside having sugar phosphate base. So it gives the nucleoside as well as the bases, sugar phosphate, and the energy. By breaking of bond, it provides us energy which is required to polymerization, to addition of this base pair, adenine, thymine, cytosine, guanine, like this. Okay. So in this way, the deoxyribonucleoside triphosphate play dual role. ये दोनों काम करता है, एनर्जी भी देता है और अपना रॉ मटेरियल भी सुगर फॉस्फेट बेस दे देता है. इसमें आप लोगों को समझने में कोई दिक्कत नहीं होनी चाहिए. The two strands, second thing the two strands cannot be separated through the entire length. अब ये replication, this replication will start where? Is it through entire DNA fragment or a part of DNA? So if it occurs in entire set of DNA, it is very We have just discussed the fast accuracy and accuracy of the DNA. So it requires very huge amount of energy if it occurs in entire set of DNA. So only a part of DNA replicates at a time, and one by one added to it progresses forwardly, and the entire set of DNA in time it takes some time to replicate. Okay, so that's why we say cycle S phase is the longer phase. So in S phase, this replication is occurring S phase of cell cycle. You understand? So the two DNA strands cannot be separated. So the portion where they separate with the help of enzyme helicase enzyme added there, and separation begins. It's unwind the DNA thread, which is a wind up with one another to form with the hydrogen bond. Now gets separated, and the portion where it gets separated is known as replicating fork. It's a fork like when they remains associated, and when they separate, the figure is just like a fork like what you are looking here in the diagram. No problem to understand. Okay, it's a DNA strand, and when helicase enzyme is added, it makes it separated, and then the dependent DNA polymerase, because polymerization of DNA is going to be happen, not RNA. DNA dependent, remember, DNA dependent DNA polymerase replication brings about replication, while DNA dependent RNA polymerase brings about transcription in messenger RNA. So later on we will study next topic. Okay. So DNA dependent DNA polymerase catalyzes the reaction. Second thing, the replication of DNA. You are just observing this replication form. Here is 5 to 3 direction. One is 3 to 5 prime direction, anti-parallel direction. The DNA segments, the strands are there. So always the replication begins in, remember, 5 prime to 3 prime direction. Are you confused about the 5 prime or 3 prime? These are the prime is here is the number of carbon. Remember, in my previous class I have already explained about the DNA structure. 3 and 5 are the position of carbon in ribose pentose sugar. नहीं जानते होगे तो देख लोगे, उसको फिर से समझ में आ जाएगा. So 5 in one from 5 to 3 direction, always transcription. The DNA polymerase catalyzes in only one direction. Remember, never forget. 5 to 3, बड़ा से छोटा की तरफ ऐसे याद करोगे तो भूलोगे नहीं. 5 to 3 direction, it's always catalyzed. Another thing, the two strands are catalyzed, replication occurs not continuously. Again, it's required very huge mass of energy at a time that cannot be provided by this cell. है ना? जो अब साधारण अब तुम 6 वोल्ट के बैटरी से तुम एसी नहीं चला सकते हो, तो एनर्जी सेल को जितनी एनर्जी है वो उतनी ही इफिशियंट है कि वो वन डायरेक्शन में कैटलाइज कर सके, और इन अनदर डायरेक्शन इट इज डिसकंटिन्यूअस. So here you know that the thread is the DNA template. Both the strands, what type of template? They are going to replicate. They involve in formation of new ones. So they are called template. So here it is written as template strand, parental strand both. This is one 5 to 3 prime direction. This is 3 to 5 prime direction, anti-parallel. So transcription here replication occurs in 5 to 3 prime direction, in again 3 to 5 prime direction. Okay, the thread is 3 to 5 prime direction. The direction of replication is always from 5 prime to 3, which is anti-parallel to the parental template strand. So never be confused. If it is, ऐसे समझो, यदि ये 3 से 5 डायरेक्शन में है तो रेप्लिकेशन हमेशा 5 से 3 डायरेक्शन में होता है, इसलिए इधर कंटिन्यूअस होगा. और इसका कॉम्प्लीमेंट्री स्टेट 3 to 5 प्राइम में होगा तो वहां रेप्लिकेशन अगेन उल्टा डायरेक्शन. देखो ये रेप्लिकेशन इधर से हो रहा है तो ये रेप्लिकेशन इधर से हो रहा है, क्योंकि अगेन 5 to 3 प्राइम डायरेक्शन में रेप्लिकेशन होना है. सो दिस वन अकर इन 5 to 3 डायरेक्शन discontinuously. It means discontinuous replication occurring 3' to 5' direction of the template strand. Here in this figure. These are the newly synthesized. These two are the newly synthesized DNA. So here you can see the two strands are there. Here also two strands and two strands. So from one DNA strand, two DNA strands are formed, and they are equivalent, equal to that one. Only the difference is the semi-conservative. One of the parental genome and another is from the nutritive medium added. So here we are seeing. Now the time we have already discussed that during S phase of cell cycle, if the cell division doesn't occur and DNA replication occur in S phase and cell unable to fails to divide, then there will be lead of polyploidy. Means the cell contain more than one set of chromosome. Like a haploid, one set of chromosome, diploid, two set of chromosome, triploid, tetraploid, pentaploid, hexaploid, octaploid, like this. These all are called polyploid. Entire set, 23 human being में जोड़े तो वो बढ़ता चला जाएगा. So in this way, polyploids are formed in this way. Now, when in another, there is another problem in this continuous synthesis of DNA replication in again 3 to 5' direction. Replication occurs in 5 to 3' direction only. So these continuous fragments are joined by another set of enzyme known as ligase. DNA ligase enzyme join them further and make the complete two-strand complementary to their parental strand. These are the parental strand and these are the newly synthesized strands. Whatever the original paternal strands are there, they are accordingly. If they are added in thymine, here again adenine comes. Here thymine comes. Thymine searches adenine. Adenine searches thymine. So adenine thymine earlier and adenine thymine later. So same DNA genetic materials are to be synthesized. Okay, this is very efficient. Never folded. 2000 base pairs are synthesized per second, which is imagine, सोचना, इसको दिमाग से सोचो कितना फास्ट ये हो सकता है. तो in this way we have discussed the DNA replication, how it's occur, and replicating form. Many times you will see that in board examination this question is very important. Okay, so we'll study not only in board examination, in NEET examination also. Okay, the speed, never forget this one, the speed, number of DNA and the dual nature of this one, the work of DNA ligase, another things. I forget to explain this. Replication is not occur in a haphazard manner anywhere. It's required a specific site where the replication begins. That site, this time this question was asked in NEET. So that site is called ORI, origin of replication. In the entire DNA set, if there is a origin of replication, the replication begins from there. So in making rDNA, recombinant DNA technology, the ORI is not available, then ORIs are given by the vectors. So vectors, plasmid vectors, play important role to provide ORI. How many number of origin of replication ORI there in the vectors, so many copies of DNA will be produced. So that they play very important role in biotechnology or DNA technology. Okay, thank you.